Our project was to model the synovial joint on a microchannel chip. Our client wanted us to model the synovial joint on a microchannel chip that would enable osteoarthritis drugs to be tested. The chip would also have to accurately mimic how bone, cartilage, and synovium would react to a drug in vivo. Our device works on basic microfluidic device principles. Microfluidic devices have channels that have cells or liquids in the channels that are injected or removed with tubing, syringe adapters, or holes. The flow can be controlled passively via hydrostatic pressure or actively through external systems such as pumps or pressure controllers. The channel can model tissue types by changing design parameters including dimensions and flow rate. Osteoarthritis is a degeneration of cartilage in the underlying bone. It is common from middle age on and causes pain and stiffness in the affected joint. Approximately 50 to 60 million people are affected in the U.S. There are no proven therapies or treatments, which is most likely due to incongruence between in vitro and in vivo models. Most osteoarthritis models focus only on cartilage instead of the whole joint.
The synovial joint is composed of cartilage, bone, and synovium. Each have their own unique composition. Cartilage is composed of collagen type 2. It is highly hydrated, viscoelastic, and has an isotropic matrix which encapsulates chondrocytes. Bone is composed of stiff collagen type 1, and it's a woven calcified structure that encapsulates osteoblasts, osteoclasts, blood vessels, and nerves. The synovium is composed of scattered blood vessels, fat cells, fibroblasts, macrophages, and few lymphocytes. The structure of synovium can vary based on the subintimal layer. Each component of the synovium joint requires their own mediums with unique properties. For the osteoblasts, mostly basal media is used with osteoblast growth supplement, fetal bovine serum, and some sort of antibiotic solution. For chondrocytes, DMEM media is used with insulin, transferrin, and fetal bovine serum. The synovium will use hyaluronic acid and lubricant to more accurately model the synovial joint secretion. With our project, it is necessary to stimulate osteoarthritis inflammation, so for this, we supported our project with some available research. We found in this paper shown on the slide that it's possible to use the inflammatory cytokine interleukin 1b to induce osteoarthritis inflammation in cells, which occurs when the cytokine is introduced into osteoblasts.
The diagram on the left illustrates the basics of our optimal design. The design consists of 3 distinct cell chambers, one for each tissue type. Each chamber has a corresponding channel that will deliver cell-specific media as well as inflammatory cytokines to induce OA inflammation. We place membranes between the chambers to keep each media isolated. However, to facilitate cell-to-cell communication, we created micro-sized pores within the membranes next to each hydrogel. In order to best simulate in vivo conditions, we created a central channel through the middle of the chip. This channel will act as the blood flow found in vivo and will be how the drug is introduced into the chip. Each design choice made was intentional to accurately model in vivo conditions and optimize the effectiveness of the chip. The dimensions were chosen to maximize the length and width of the chip in order to maximize the cell-to-cell interaction surface area. PLA filament was chosen as a 3D printing material because it is biologically inert and therefore would not interact with any other materials in our design. Hyaluronic acid hydrogel was chosen to seed cells into because it has optimal cell adhesion and is found naturally in the body. The central channel only has pores in the osteoblast and synovial chambers to mimic the avascular nature of cartilage in vivo. In order to accurately model the amount of drug each tissue would receive in vivo from the bloodstream, research was done into the porosity within bone and synovium. We found that osteoblasts have the average pore size of 100 nanometers, and synovium has an average pore size of 24 nanometers, which are the pore sizes we use in our design. Each media introduced into the channels will have a flow rate of 1 microliter per second. We determined these flow rates to be optimal based off of previous research done on microfluidic devices. This slide contains an image of our SolidWorks file. The purpose of the slide is to show you what our final design looks like. The chip was created to become a solution for the incongruence between current in vitro testing and in vivo conditions. We were able to achieve this through all of the design specifications talked about on the past couple of slides.
The cost of one device would be just under $100, including the PLA for the device itself and the hydrogel to fill the chambers. This is relatively inexpensive, however, it does not include the cost of pumps, cells, and culture media. The global market for osteoarthritis drugs is almost $6 billion. Pharmaceutical companies spend millions of dollars on clinical trials and billions of dollars in the whole process of taking a drug to market. This device could provide more accurate in vitro testing before advancing drugs to the clinical trial phases. This would greatly reduce the costs and time involved in drug development, including costs of failed clinical trials. Future directions of this project include using the device to culture cells and eventually inducing osteoarthritis pathology and testing drugs.